So far in this course, we have reached messenger RNA, and now we're going to do something with it. Today we're going to translate that mRNA into proteins, protein synthesis. Now, this isn't just a lecture on protein synthesis. As I've told you before, viruses are parasites of the host translational apparatus. Yet, it wouldn't be very interesting to tell you how cells translate mRNAs unless viruses did something different. And of course you do. And that's what we're going to be talking about today. How viruses translate a bit different and how they modify the translational apparatus for their own means. So here we are in the Baltimore scheme. We have traced all 7 genome types to mRNA, that green molecule in the middle. And today we are going to take that mRNA and see how the cellular translational apparatus uses it as a template for protein synthesis. So first, let's review the basic translational apparatus of the cell to make sure we are all on the same page. Here on the upper right is a typical messenger RNA. Cellular mRNAs look very much like this, and so do many viral mRNAs, but not all of them. A typical cellular/viral mRNA has a 5' cap structure, which we've talked about before. It's put on during transcription of the mRNA, and there's a structure of the cap. It is basically a G linked in a 5' to 5' linkage to the first base of the RNA. And this cap is found on most eukaryotic messenger RNAs with the exception of certain organelle and viral mRNAs. Some viral mRNAs do not have caps. And as we said, it's a 5' 7-methylguanosine joined to the second nucleotide of the RNA by a 5' to 5' phosphodiester linkage. And this cap is important for many aspects of mRNA biogenesis and function, including transport, turnover, and translation, as we will talk about today. mRNAs are also composed of 5' and 3' untranslated regions or non-coding regions, a 3' poly A tail, and an open reading frame, which typically encodes a protein. The 5' untranslated region is typically 50 to 70 nucleotides long, although it can be much longer. It often contains secondary structure, and this is an impediment to ribosomal transit. It must be unwound to allow the ribosome to move from the 5' end to the initiation codon. So the length of the UTR and its secondary structure most certainly influence translation efficiency. Then we have our open reading frame, which typically begins with an AUG, but not always, as we'll see, and then a stop codon, followed by a 3' untranslated region, which can also regulate the efficiency of translation initiation and mRNA stability, and a poly A tail, which is also necessary for efficient translation. Translational machinery consists of ribosomes. These are large protein RNA assemblies, of course. The eukaryotic ribosome is shown here, are flying turkeys. They consist of 40S and 60S subunits. Each is made up of ribosomal RNA, one or more, and quite a few proteins, as you can see. And the RNA is an important part of the ribosome. Here on the right, it's shown in pink. The protein is in blue. And there is good evidence which indicates that if you take away the protein, the RNA can still carry out the catalytic function of the ribosome, that is to make a protein, to catalyze peptide bonds. tRNAs are another important part of the translational machinery. These are the adapter molecules that read the code on the mRNA and put in the right amino acid. And here are 3 views of tRNA structure. On the left is the linear sequence of different bases forming this tRNA. You can see the anticodon is at the bottom with the red box around it. This is the part that reads the mRNA triplet. And the amino acid attachment site is at the other end at the 3' end. In the middle is a alpha carbon trace of the tRNA. Again, the anticodon and the AA attachment site are highlighted. And finally, a space-filling model. So these are L-shaped molecules, you can see. Other materials are needed to translate proteins. These include initiation, elongation, and termination proteins. We tend to divide the translation of an mRNA into 3 stages: initiation, elongation, termination, and there are proteins needed for each step. And in eukaryotes, these are called EIFs for eukaryotic initiation factors. Elongation proteins, eukaryotic or EFs, and termination proteins are ERFs. Now, in eukaryotic cells, we now recognize 2 distinct mechanisms of protein synthesis initiation. There's 5' dependent initiation, which is done on most mRNAs, and in this mechanism, the initiation complex binds to a 5' cap structure and then scans to find the initiation codon. And up until the 1980s, it was thought to be the way that mRNAs were translated. But viruses lead the way. Internal ribosome entry was discovered on viral mRNAs. This is when the initiation complex binds near the initiation codon. It does not bind at the 5' end, as does the ribosome in the other mechanism. And this was initially discovered in viral genomes, but now is known to occur in cells as well. So let's take a look at 5' dependent initiation. Here we have a 40S subunit. These are free in the cell. They associate with a variety of initiation factors that are important for these initial steps, including EIF1A and EIF3. And then a ternary complex comes and binds. The ternary complex consists of GTP, EIF2, and the MET tRNA. Ternary complex, as you will see, plays an important role in initiation and in the regulation of that process. It associates with the 40S subunit. It then binds EIF4G, which is this red molecule, which then binds the cap. Now, we don't know if this is exactly the sequence of events, but this is just one version that I'm presenting to you. Now, the key here is how this initiation complex, you can see this one is called the 43S pre-initiation complex. How does it recognize the mRNA? Well, it does so in this mechanism by virtue of the 5' cap structure. This little blue box with a C in it, that's our signal for a cap structure. There is a protein in the cell, a eukaryotic initiation factor called EIF4E, and it binds very specifically to the 5' cap. EIF4E in turn binds to EIF4G. You can't see that interaction here. It's covered up by EIF2, but you'll see it later. EIF4G in turn binds EIF3, and then EIF3 binds the 40S subunit. So that brings in the 40S subunit along with all the other factors in the pre-initiation complex, the ternary complex, etc. Okay, so that nucleates the initiation complex at the 5' end of the messenger RNA. So the cap is very important for this, and that's why this is called 5' end-dependent initiation. EIF4G is a large protein, 220,000 Daltons molecular weight. It has, as shown at the top here, a variety of domains for interacting with other proteins, including poly-A binding protein, PABP, and we'll look at the function of that in a moment. EIF4E binding site is here, EIF3 binding site, and some sites for others as well. This 4G is cleaved by viral proteases from picornaviruses, and we'll talk about that later, but here is the cleavage site for the protease. Now at the bottom is a examination of the interaction of 4G with this initiation complex, and we've taken away a lot of the other factors so you can see it more clearly. And again, we have the mRNA with its 5' cap structure. It is interacting with EIF4E, and EIF4E, of course, binds to the cap. EIF4G binds to EIF4E, and EIF4G binds to 3, which in turn binds to the 40S subunit. So you can see how the 40S is brought here by these series of protein-protein and protein-cap interactions. All right, so now we're going to move to the next step in translation to find the AUG codon. So here at the top we have our 48S initiation complex. It's all of these factors bound to the mRNA via the cap, and the point is to find the AUG, where initiation is going to begin. And it's believed that this complex, the 40S subunit and associated factors, has to scan or move along the AUG to initiate translation, and that may be an energy-dependent process. And once the 48S complex finds the AUG, then translation initiation can begin. A series of events then occur. EIF5 comes in and helps to hydrolyze the GTP in the ternary complex. A lot of initiation factors are released. And then the 60S subunit can join to produce this complex, the mRNA, the ADS ribosome, and the MET tRNA initiation tRNA. Now, in order for the initiation complex to move to the AUG, it has to make sure that there is no secondary structure. So on the top here, we're showing one such secondary RNA structure, which could impede the transit of the initiation complex. The complex has built within it an RNA helicase, and that's this green protein here, EIF4A. Its function is to melt out any secondary RNA structures that are present in this region. Now, this helicase is only good up to a certain point. If these structures are very, very stable, it can't melt them, and then translation is inhibited. But in general, the amount of secondary structure can serve as a regulator for how efficient the mRNA is translated, and that's because the secondary structure can regulate the transit of ribosomes to the initiation codon. Now, we believe in general that the mRNA forms a circular loop in order for efficient translation to occur. And the way that happens is shown here. The N-terminus of EIF4G, in addition to having a binding site for EIF4E, also has a binding site for the cellular protein PABP, poly-A binding protein. And that, of course, as its name suggests, binds the poly-A sequence on the mRNA. It also interacts with the N-terminus of EIF4G. And this allows the RNA to form a closed loop. And biochemical and genetic evidence indicates that this closed loop is necessary for efficient translation of an mRNA. If you disrupt the loop genetically or biochemically, you get less efficient translation. And we're not sure why a closed loop is needed for efficient translation, but one idea is that as the ribosomes translate the open reading frame, they reach the stop codon, they can simply keep moving on the mRNA and reinitiate. And this also is one reason why the poly-A tail is important for efficient mRNA translation.